Hello and welcome to my channel ominibioentrance, and today we will be discussing CSIR PYQs June to 2023 Shift 1. In Shift 1 I will be discussing applied biology methodology, DNA replication, protein synthesis and genetics. Questions from both part C and B will be discussed. I have divided into two parts. This is the first part. Coming to the first question, these questions are from part B. Consider alleles capital A and small a in a population. The frequency of heterozygotes will be highest when. So if you see this graph, you can see that this is the heterozygous graph and the frequency is highest when P value and Q value are equal to 0.5 each. So when the frequency of a capital A is equal to frequency of small a is the answer. So answer is option 3. Then next one. Recessive lethal alleles are never completely eliminated from the population because now deleterious recessive alleles are recessive alleles that can cause disease in an organism. If an individual is heterozygous, they inherit one recessive allele and one dominant allele. And the dominant allele, it will mask the expression of the recessive allele and the individual doesn't show the deleterious effect. Thus, the heterozygous individuals allow recessive allele to be present in the population. So, recessive lethal alleles are never completely eliminated from the population because they are maintained in the population as heterozygotes. Answer is option 4. Then next one. Which of the following factors will not have any impact on the resolving power of a bright light microscope? So, wavelength of light. Blue light has shorter wavelength and red light has longer wavelength. So, it is dependent on wavelength of light. Then refractive index, that is medium between objective lens and the specimen. It is also dependent on that. Angular aperture. So it is dependent on 3 factors except intensity of light. So the answer here is option 2. Then which of the following terms represent different types of methods in phylogenetic tree construction? Select the option that represents all distance-based methods. So unweighted pair group method using arithmetic average. That is correct. Minimum evolution method, ME method. That is also a distance-based method. So the answer here is option 1, that is A and B. Then next one. Which one of the following mRNAs is most likely to be exported out of the nucleus? Spliced RNA associated with poly A binding and cap binding complex. That itself is the answer. Misspliced RNA, that was not possible. It will not be exported out. Spliced RNA associated with spliceosomal complex, no. Uncapped and unspliced RNA, that also will not be exported out of the nucleus. So the answer here is option 1. Then, which of the following types of promoters would not be used within the tDNA for expression of a negative selection marker gene for generation of transgenic plants by agrobacterium mediated transformation? So, a negative selection system, it is described in plants for the genes that result in the death of the transformed cell. Now, if it is a strong constitutive promoter, then the whole plant will die off. So naturally you will not use a strong constitutive promoter. So the answer is option 1. Then next one. Segregation of alleles can occur either at anaphase 1 or anaphase 2 of meiosis. Which of the following is an ideal model system for identifying the stage at which allelic segregation occurred? So this first division segregation, second division segregation we study in Neurospora crassa. So Neurospora crassa will be an ideal model for identifying the stage at which allelic segregation occurred. So the answer here is option 3. Then next one. Which of the following statements about lines present in the human genome is true? Lines preferentially localized to AT-rich region. That is correct. Lines cannot transpose independently. They can transpose independently. So 2 is not correct. By parasitizing on the sign element transposition machinery, lines can attain very high copy number. Signs are not autonomous. Retro elements are dependent on the lines for transposition. So lines will not parasite on the sign element. The ALU family is the most prominent line family in terms of copy number. It is the L1 family, line family, that is most common. So the answer here is option 1. Then next one. Which one of the following statement regarding molecular markers for genotyping is incorrect? Polymorphism in intronic region of a gene cannot be used for trait mapping. Now, intron polymorphism, they are highly efficient and easily examined molecular markers located adjacent to exonic regions of functional genes. And this may be associated with targeted trait variation. So, the incorrect statement is polymorphism in intronic region of a gene cannot be used for trait mapping. It can be used for trait mapping. So the answer is option 1. Then next one. Which one of the following translation factors is used at the step of translation initiation and defined as anti-association factor for 30S and 50S subunit interaction? So IF3, initiation factor 3, it prevents 30S and 50S subunits from combining prematurely. So the answer here is option 3. Then next one. Which one of the following codons is used to code for solenocysteine in Escherichia coli? So it is UGA, which is used to code for solenocysteine in Escherichia coli, and solenocysteine, it is the 21st amino acid in the genetic code. So the codon is UGA.